Okay, so good morning once again. And you have come here for this population genetics school. So as you know, I mean, in the next 2 weeks, you will hear a lot of exciting, interesting problems about evolution. So of course, all of you know what evolution is. At least you have some rough idea. And population genetics is simply a framework to understand evolutionary processes, evolution, how it works, its dynamics, and so on. Okay. So in the kind of question that one asks is, in population genetics, the kind of question one asks is, how does, let's say, the genotypic frequency change with time? How does phenotype change with time? Under the action of some evolutionary forces or processes. So first question from you, what are the evolutionary forces? When you think of evolution, what are the first words that come to your mind? You have to speak because, otherwise it's not nice. So when you think of evolution, what are the processes or forces that you first think of? Yeah.
Just a quick announcement. Please use the microphones when you ask a question or give answers because it's being recorded. Yeah.
Anybody who doesn't answer doesn't get lunch. So, everybody's laughing. That means you had a good breakfast, which means you have energy. So, what is the first thing that comes to mind whenever you read in school about evolution? Huh? Selection. And do speak in that. Yes. Selection. Good. What else? Mutation. Yes, very good. What else? Recombination. Good. Okay. I hope it is large enough for everybody to read. Selection, mutation, recombination. Never heard of that. Okay, what else? Migration. That's it? Okay, I think it's a long list anyway, right? Okay, so what we will do is so I will start with genetic drift. It's about stochasticity, okay? So I will speak a little bit about that. I will introduce some very, very basic concepts here. And then I will probably add mutation to it today, if there's time. And tomorrow, then I will add selection. And it depends on how it goes. So we will, I don't have to cover everything that I have, planned. But, of course, you have to answer questions and ask questions, and then we'll see. Okay? Okay. And while we are discussing these things, I will give you small exercises which you can take home and think about, and we will discuss them perhaps tomorrow. Okay, so please pay attention. Okay, so I want to speak about random genetic drift. Okay, so it's about stochasticity. And whenever I think of stochasticity, I mean, it's easier for me to think of a much simpler model. So before we come to evolution, I will ask you to play a little game with me, and it's called Gambler's Ruin. Why gambler? So as some of you might know, probability or stochasticity, I mean, start to think about these ideas. As far as I know, it has roots in gambling, games of chance. And this started around 1600s or so. And there was this correspondence between these two famous scientists, Pascal and Fermat. So I think all of you have, again, heard of these two guys, right? Pascal, anybody who has not heard of Pascal? Pascal? Pascal's triangle in school we had done. Fermat. Everybody has heard of Fermat? Fermat's theorem, last theorem, right? Okay, fine. So this is the simple model that we will first discuss, and then we will come to evolution. Some of you have seen this before with me, so please keep quiet. Others, please think about it. So here's the game. So let's forget evolution for a second. So all of us are going to play this game. We have a fair coin in our pocket, a 1 rupee coin. Fair means the head and tail are equally likely to come. Okay? And you have 50 rupees in your pocket. So when you toss a coin, if you get a head, you win 1 rupee. If you get a tail, then you lose 1 rupee. And you're starting with 50 rupees in your pocket. And the game will stop if you get 0 or 100 rupees. So you have to keep playing the game until then. Are the rules of the game clear? Right? Toss a coin, head, you win 1, tail, you lose 1, and you have to keep playing till you get 0 or 100. Okay? And I'm asking, what is our fate? And you have to keep playing, right? So what's your fate? Each of us, that is. I'll give you some options. The game never stops. That means you don't leave this room. Or game stops and you leave with 0 or 100. And this is my favorite answer. I don't know. Okay, so we'll take 2 minutes and think about it, because this is a really, really, really basic thing. And once we have got the funda right, I think things become a little bit easier. Okay, so I'll give you 2 minutes to think about, and of course, feel free to chat with each other. Chat meaning discuss with each other, and then I will come back and talk to you. Okay, so start. Feel free to chat with your neighbor. This is not an exam, right? Okay. So time starts now. Yeah, done? Okay. Okay, so let's take a poll. How many of you think that we will never leave this room? Please raise your hand. Okay, good. So one person says we will never leave the room. Okay, how many think we will leave with 0 or 100 rupees? Oh my God, almost everyone. How many think I don't know? Okay, fine. Okay, so that's very amazing, unless you heard the previous lectures. Because when I started to think about this problem, I always thought the first option, that we will not leave. So maybe some of you want to tell me, why do you think we will be able to leave? Because this was very unintuitive for me when I started to think of this. So all those who raise hand, one of you raise hand again and tell me, why do you think the B option, it's the correct option, by the way. So can you explain in your own words? Yeah. Why does it have to get? So I'm here, 50 rupees, in the middle of this room. I toss a coin, it's a fair coin, right? So I get a head, maybe another head, tail, sort of equally chance. So, yeah, but will I get there? Won't I be just doing this? No? Isn't that what you would think? I mean, maybe you don't think that way, but at least that's what I thought. What you're saying is right. So at least that's what I thought, that if I plotted, so I did this experiment in the computer, and the amount that I have in the pocket, I did 50 rupees. So I thought that, this is a fair coin, chance of getting a head and tail. So I win something, head, then tail, tail, I lose back, head, tail, I will keep doing. And when I ran this in the computer, this is what I got. So indeed, my number kept fluctuating, some ways, okay? Like that. For a long time, I stuck close to 50 rupees. So I will plot you exactly what I found in the computer. So I got something like, around like 800 tosses or something like that. It stayed like that. And then, okay, let's say 500. And then some funny thing happened in which I got a string of heads, okay? And that again sort of hanged around. And then maybe I got something, then tail, back, and heads, and so on. And then it got about 100. Is that what you would have expected intuitively? It's a fair coin, and I'm starting right at 550 rupees, right? So what happened here? This is what we expected intuitively, that we will just hang around in the middle, okay? And the thing is that if we waited long enough, then a string of heads came, a string of tails also could come. So another time when I ran this experiment, it did like this, and then at a different instant, it went to 0. So this is very different from what at least I had expected. I had expected for a long time it will behave like this, but there's a large fluctuation from a way which is different from our intuitive expectation, right? So which is very, very different from what we expected at short times. So what is this thing business about short times and long times? I asked you, okay, eventually whether we leave or not, but somebody said long times. There is this time coming into picture in this. How is time important in this? By time you mean the number of tosses. That if I do lots and lots of tosses, then, see, first of all, it should be clear that once by chance, I hit 100 rupees. That's by that wall, and 0 rupees is that wall. By doing this dance, I wind it up, I hit that. I certainly cannot, I have left the room. That's it, finished, right? So the question is, will you hit it in some finite time, right? Okay, so now you said that, long enough time, it would happen. So why is this long enough coming into picture? Can you give me some sort of understanding of that? Yes? Go ahead. Yeah, can you say louder? I can't hear it. Okay. Okay, so I'm not sure I understand completely, but this is how at least I understand. The thing is this, suppose I toss 2 coins, just 2 coins, okay? What's the chance of getting 2 heads? Just 2 coins, forget this for a second. Just 2 coins, if I toss, I can get 2 tails, 1 head, 1 tail, or 2 tails, right? What's the chance of getting 2 heads? 1 by 4, right? What's the chance of getting 1 head? 1 by 2, right? And chance of getting similarly the 2 tails is quarter. So chance of getting this head and tail equal number is higher than 2 heads or 2 tails. But if I do sufficient number of experiments, I will get to these rare events. That means the ones which have a less probability. So let me rephrase this. So when I'm thinking of this, my intuition, which was incorrect, there I'm thinking of this common events, something like head, tail, head. But something like head, head, which has a lower probability, for that I have to do sufficient number of experiments. And that's what she, I think, meant. That if I wait long enough, if I do enough tosses, then I will get to these sort of events which have a low probability. And therefore, if I keep going, these low probability events will keep coming, and I'll hit 100 or 0, and that's the end of the game. So yes, we will leave the room. Is there anything special about 50 rupees? Do you think if I had started with 95 rupees, we will still leave the room? How many say the B option is still true? If I start with 95 rupees instead of 50 rupees, do you think the B option is still true? We will leave the room? Yes? Yeah. If instead of fair coin, if I had an unfair coin, which means heads come with larger chance than tails, will we be able to leave the room? Yeah. So there's nothing special about fair coin or 50 rupees. You will leave the room. Okay. But the times and the chance that whether if you start with 95 rupees, will I wind up with 100 or 5? Those things will differ. Okay. So this is a very simple problem. And I think this is sort of a nice sort of a picture image to keep in mind. That initially at shortish times, fewer tosses, you will hang around, which you might have expected intuitively. One sec. And then later on, if I wait long enough, then I will wind up with these very different things, which have large fluctuations. So when we think of stochasticity, the name of the game is fluctuation. The fluctuations will be high. Yeah, question. Yeah, sure. It could have happened. This is a possibility. This is the typical trajectory. But it's possible that I could have gotten this initially. And it could have happened. Okay, and then yet, yes, it's a trajectory. So I think there are these program simulators which you can play with, and they are very instructive. So I strongly encourage you to play with these to get some intuition into this, how this kind of fluctuation, etc. works. Okay, okay, good. So now that this is clear, let's go to evolution. And I will define for you a very, very simple model, which is the sort of very basic model. Okay. So from 1600s, let's go to 1920s. So the idea of this random genetic drift is about 1920s. Due to this, I think this Dutch couple, Hagedorn and Hagedorn. So the idea is that, like, for example, Drosophila lays about 100 eggs, each Drosophila, right? But there's only a finite carrying capacity. There's not food for all the eggs. So even if they're equally fit, right? So some of them will die just by chance. So even if they're equal, all of them laid 100 eggs. Some of them will die by chance, and that stochasticity in the offspring number is what is captured by random genetic drift. So we'll write down a simple, small model to understand that. Okay, so let me explain this point a little bit more. Suppose I just have this long genome, and I'm just looking at one locus in it, which has 2 alleles, A1 and A2. And I have just 6 individuals, simply because it's easier for me to draw things. So let me repeat this point first. Suppose I started with 3 A1s and 3 A2s, and all A1s and A2s are equally good. Okay? And I cannot draw 100 eggs of the Drosophila. So therefore, I'm just saying that let's say they're bacteria. Each of them produces exactly 2 offspring. And so on, right? Okay. And then because there's food only for 6 individuals, out of these 12 guys, some of them have to die, right? So what do you suggest? How should we remove half the population? They're equally good, all 12 of them. Any suggestions? toss a coin and then do what? Yeah, say. Yeah, but I'll find it difficult to keep exactly 6 of them. But yeah, maybe we can do that. So we toss a coin and say head, okay, live, tail, die, whatever, right? Till we get 6 of them, and that's it. Finish the game, right? Or what I would do is to take a dice with 12 faces and throw the dice with equal probability, and then basically the sort of game you are suggesting. So therefore, we will wind up maybe with something like this. So I started with a very democratic situation. All were equally good, and equal number of A1s and A2s. But just by fluctuation, we winded up killing more A1s than A2s. And then there is a small little bias towards number of A2s, right? And then maybe in the next generation, there is a slightly higher chance for A2s to survive, okay? So this sort of basic idea, we can formalize into what is called a neutral Wright-Fisher process. So let me define this model for you. So I will skip the intermediate step. And let me draw exactly what I have. So what we do is to say that, like I said, I will throw a dice. It has 6 faces, and all faces have equal chance. So let's say whatever number comes, that individual I keep. So I throw a dice. So this is my t equal to 0, the parent generation. And at t equal to 1, I say that, okay, throw a dice. If 1 comes, okay, then this is the parent. Then I throw the dice again. And I'm asking, who is the parent of the second offspring? I got a 2. Then I throw the dice again. And this time I got a 4. So then I drew this. Then again, I got a 4. Again, I got a 4. See? And then I got a 5. 3, 4, 5, 6. Yeah. Is the process clear? Is it clear? Yeah, let me repeat it. I think it was not very clear. Okay, so I started with this. So I'm saying this is, you don't have to write any sophisticated programs for this, right? You can just play this game in your head. You can just take a dice. Okay, so you have 3 A1s and 3 A2s. You throw the dice. If you get a 1, you go sequentially like this. You go to this first offspring and ask, who is your parent? Suppose the dice showed me 1. Then I connect this with this. And because there is nothing else going on, there's no mutation, no selection, no nothing. Therefore, this offspring copies the parent. If this is A1, this becomes A1. Then I threw the dice once again. This time maybe then now I'm asking, who is your parent? The second offspring, who is your parent? This time maybe I got a 2. So then I connected with the second parent. Okay. And then again, third time when I did, maybe I got a 4 on my dice. So I connected this guy with this and copied from this to A2. And similarly, again, I got a 4. So this is a string of, on my dice, I got a 1, 2, 4, 4, 4, and 5. And therefore, A2 was the parent of these offspring, and this is this guy. Is this clear? Easy to sort of run it in your head. Is it okay? Yeah? Okay, so let's think once more. So if I run this program, what do you think will happen this time? So just run this program, imagine throwing this dice and just playing this program in your head. There's nothing, there's no mutation, no nothing. That's just this what I just described to you. Let's take 2 minutes again, chat, think about it, and come back. Yeah? Okay? Please think about it. Go through this in your head. All done? Ready? Yeah. Okay. Okay, so I would like to hear from people who have not spoken, because all of us have to speak. Okay. Somebody else wants to tell me, what do you think if I run this process long enough? What do you think can happen? Yes, that's right. So it's just like the random walker problem or this coin problem that we did. Can you explain a bit more?
So it becomes A2 or A1. It's more likely to become A2 at the end as compared to A1. How I thought about if each of representatives was one offspring for the group. That means picking up from the sample, the probability of picking up an A2 is.
Yeah. So at this instant, it happened to be slightly biased, but it's just our trajectory, right? So actually, this is not going to determine what happens eventually, because it's possible in the next step, I might wind up again with 3 A1s and 3 A2s. Okay, but you're right. Eventually, just like we just talked about coin problem, just like that, if you keep doing this, so here in this case, A2 is slightly more probable, maybe it happens, okay? So this is the probability of A2s. Number of A2s here is 4. So it's more likely with a 2/3 chance that I will have A2s in the next generation. More A2s will be there, right? And the other ones will be smaller probability. So we keep doing this problem, and it's possible that either we will wind up with all A1s or all A2s. So let me draw one such stochastic trajectory for you, because I want to make a point. So just be patient. Let me draw it. And I'm going to draw the full genealogy. Okay. Now I need to draw the arrows. A1. So almost there. Okay, so I think I drew it correctly. So this is 0 generation, 1, 2, 3, and 4. Okay. So like, it so happened. So from each of these A1s, an arrow is coming out because it's a single parent. But you see, for example, this A2 produced 3 offspring, right? And similarly in the other generations. So there's a fixation which has happened. So fixation, so let me introduce this word for you, which means all have become A2, right? So that has happened at this time step. But I went for a reason, one more step, okay? Right, okay? So like as you suggested, that yeah, maybe all become A2. So this is a concrete configuration where I'm showing that it could have happened, right? Okay? So therefore, one more question that we are asking is, what's the chance? So as you said, that it could be all A2s or all A1s. So therefore, now we can ask a quantitative question. What's the probability that all will become A2? So more precisely, the question is, suppose we start with little n number of A1s or A2s in a population of size n. And what is the probability that eventually all are A2? I have population of A2. So this is called probability of fixation of A2, that is, right? Okay. So then you can do some calculation and so on, but we will not do that. But there's a simple way of seeing this, and that is to track the genealogy back. So you see what you have here is there are all A2s. Okay. So let's go back in time. And so this one, these are two offspring, and this is the parent. Okay, that's the yellow. Here, these are the yellow, and so on. Right? So therefore, this is the one that we have. And you see, I will draw this guy, and this guy, and this. So look at the yellow ones. These A2s, their grandparent was A2. Right? Similarly, look at this A2. What happens here? Here also, it went to that A2. This A2 same way, and same for this. And that's it. Yeah. So all A2s, look, I hope you can see the yellow arrows. Okay. So all A2s, I think I missed something about this guy. Okay. Yeah, I think it's this one. Okay, so look at the yellow arrows. What has happened? All of them able to track their ancestry to one individual. So this is called the most recent or the common ancestor. And if you look again down in time, you see it's branching like a tree, right? So it's called a coalescent tree. Coalescing because, these branches are coalescing, they are merging. If I look back in time, upwards in time, and they're branching out when you're going forward in time, right? So it's called a coalescent tree. I know very little about it, but you will hear more about it in the next week. I think Aurelian and Yun Kitano, they will speak more about it. Okay? So, but this we can use to answer the question that I wrote over there. So the question was that if I started, let's say in this configuration, what's the probability that that A2, finally, I'm claiming here that if I run this process long enough, all the A2s, suppose they became all A2s in this case, all of them will be able to track the ancestry to a single A2. So in this configuration, this A2 happens to be the ancestor. Could have been this or this, right? And these are all, there's only equal likelihood. Everything is equally probable. Okay? So therefore, what do you think is a chance that that A2 is the common ancestor of these 6 guys? They're all equally likely. They're equal chance, right? 1/6, right? Of being this. But for this A2, or this A2, or this A2, what's that chance? This A2 being is, 1/6 to be a common ancestor, provided, of course, they became all this, right? Okay. But it could have been this one or that one. What do you think is that chance? So there can be either of them could have been. That probability is 1/6. So let me write it, and again, you can try to answer this question. So let me look for this guy. Probability, any of the 6 is MRCA, is 1/6. The fair coin, right? They're all equally good. But how many are there? It could have been this one, or this one, or this one. So this is enhanced by a factor of 3, right? Therefore, this probability is half, which is you could have just guessed it, right? Is it okay? Yeah? Okay? Yeah. So it's 1 by 6 into 3. MRCA is this guy, the most recent common ancestor. Okay, yeah? Is it okay? Is this a simple argument okay with everyone for this? Okay, if it is okay, you should answer that question. So can you answer this question for me? Okay. Okay, let's take 2 minutes. Start chatting, thinking. 2 minutes. So instead of 3 of A2s, I have some little n of A2s, and the total population size is capital N. And I'm asking what's the chance that eventually all are A2s. Same argument you have to use. Just think a little. It's not, doesn't require too much work. Question is clear, right? Just generalize that. It's helpful to think in these slightly abstract ways. It's very helpful for thinking. Try it, please. Just generalize this. Okay? Anybody want to give me an answer? Yeah? Huh? Small n minus? Small n, yeah. So that's correct. So this probability, which we call fixation probability, is small n divided by capital N. Would you like to explain why? Yes. Yeah, so either of the parent could have been an ancestor, right? And that A2 is the ancestor is the number of A2s in the beginning, right? So therefore, it has to be little n by capital N. So let me rewrite it like this. Anyone could have been ancestor. That chance is 1 by n, and it's enhanced by the number of A2s we have. That's little n by capital N. Okay, that's formula. Does it make sense? If I have a very large population, so let's think of little n to be 1 for simplicity, or okay, let's say little n is 1. So it's saying that if I had a single A2 and everybody is A1, okay, in a large population, n is large, maybe, I don't know, 100, a million, the chance is very low. Does that make sense? Or maybe it's wrong. Does it make sense? Is it what you expect intuitively? Explain, can you explain in your words? Does it make sense? There's this one guy, it has to fight off all these million guys to, to spread in the population. It's as good as all the others, but it's a single lone warrior, and it has to fight off all the guys, right? So it makes sense that if larger the population, smaller is the chance of that single mutant to spread. Agree? Is that agreeing with your intuition? Similarly, if I have more mutants in the same size population, again, I expect they should have a higher chance of spreading. Okay. Is there any, looking at this answer, could you say, so this is about fluctuations, as I said. The stochasticity business is extremely hard. I make a lot of mistakes all the time about this. I mean, I always get the answers first time when I look at it wrong, because they are difficult. They're very counterintuitive many times. Okay? So now, but it's useful if I can ignore stochasticity. Do you think at least I can, stochasticity would be negligible? Can you think, looking at this answer, when it may be negligible? Huh? Raise hand and speak up. Yeah? Louder? Large population sizes. Why is that? Explain in English. Yes, speak up. So no, I'm saying this, can you explain words? Why is that intuitively correct? So he's saying that in large populations, stochasticity or these fluctuations are negligible. Yeah, but like, I don't know what sampling is. Can you say it in something more wordy? Anybody wants to say it a little bit more? It's right what you're saying, but like something, some other perspective maybe you can give. Yeah? Okay, something of that sort, yeah. Yeah, go ahead. Mm-hmm. Yeah, it's something related, something like that, yeah. So this is, he's talking about, you know, when your teacher tells you to do more experiments. Why should I do more experiments? Because the relative fluctuation decreases with the number of experiments. So I think that's what you were also saying, right? Yeah, so here, you know, if n was truly very, very large, it filled the entire room. It was 10 to the power, you know, I don't know, 30, 40, whatever number, large number you can think of. What do you think this single guy it will not survive, right? So that is called, and this fluctuation and all not going to help. So those are called deterministic theory. And what we do is to take n to be literally infinity. So it's a class of theories which allows us to forget fluctuations. So these are, don't worry too much with these words, where you can neglect fluctuations. I mean, there's no fluctuations. Literally, there's no fluctuations. And what you're dealing with, therefore, is only about mean, about the averages. And just generally, sometimes it's a good starting point. But of course, when you have fluctuations, things can become very interesting. Okay, so just keep in mind, because I think some of the speakers will say more about that. Okay, homework problem number 1. So you gave me this answer, little n by n, when I had this population of size capital N. Suppose my population is expanding. That is, I started with 6 individuals. In the next generation, I don't have 6, maybe I have 8. Let's say it's increasing linearly. The next generation, I have 2 more added and so on. So it's, or you know, in some ways it's expanding. So I started with 6, then there was 8 individuals and so on. Total size of the population is increasing. Okay, so you can think about it later, we'll see it later. Now I'm asking the same question. So start with this in a population whose initial size is n naught, initial number of A2s is also n naught, but this population is increasing with time. And I'm again asking, what's the chance that eventually we will have all A2s? Okay, so it will be a test of if we have actually learned something. Okay, happy? More or less. Okay. Okay, so much about theory. And there is this experiment that I want to describe because it's very nice. So this is an experiment by this guy called Buri. And again, it's just about drift and just to see how these concepts might play out and introduce further new concepts. So let's look at that. By the way, erasing have water or something. Okay, I want to describe this experiment to you. The name is Buri. I don't know how it's exactly pronounced, but that's what I'm going to call it. In 1956. Okay, it's a nice experiment. And so far, I forgot to say, in the previous discussion, I assumed things were haploid. Okay, I'm now going to switch to diploids and sexuals because we are going to think about Drosophila. So he considered this population of Drosophila melanogaster, and they have, and he is looking at the eye color. And this is what is known. So eye color is a phenotype. And it's known that, and you can find it in some ways. So if they have this mutation, BW75, okay, let me first write the other one. No, so this is the wild type. Its color is, eye color is red. So this is the the homozygote. But if the homozygote is BW, then the eye color is white. And if you use crossing, then you have orange color. Okay. Okay, so what he did is the following. He started with a population of all heterozygotes. So this is the orange population. So start with a population of 8 males and 8 females, and they're all orange-eyed. That is, they're all heterozygotes. Okay, and then they're mated, and about 100 eggs, as I said, were laid. And then, regardless of what's the eye color, so I forgot to say. So this eye color is neutral. That is whether the this color is red, white, or orange. They have the same fitness. They produce the same number of offspring. Okay. So now, regardless of what the color of the eye is, each of these, so there are 8 males and 8 females. Each of the females laid about 100 eggs. So you have 800 eggs. So from that, he picked, regardless of the eye color, 8 males and 8 females. So let's do this, mating, and then pick 8 male again and 8 female, regardless of eye color. Okay. And then, and then repeat this, go back, and then keep repeating it, made the new 8 male and 8 females, and keep doing this. And he repeated this process for 19 generations. And it's amazing for me, at least. He did 212 experiments, independent ones. Okay, and he recorded the number of BW75 allele as a function of time. Okay, so that's the experiment, is what I understand. So initially, and then he's recording this. So let's try to guess what he might see. So 0 generation. So he's starting with all heterozygotes, and I'm going to plot. The x-axis is the number of BW75 allele, and the y-axis is number of experiments. Okay, so what's the minimum number of BW75 allele you can get at any time? 0, right? Because 0. Okay, so that's 0. What's the maximum BW75 alleles possible in this population? All are this guys, right? Red-eyed. So therefore, how many alleles are possible? How many? 32, right? 8 males and 8 females. So, and each of them is going to give 2, right? So my x-axis and y-axis has maximum this here, and 16 is what he's starting with, because we're starting with heterozygotes, right? So initially, if I plot a histogram, it has this single thing at 212, right? That's what we will get, right? Okay. So now we do this experiment. What do you think he might be getting? Just guess, I don't know. So it's like, the 212 experiments is like this kind of trajectories I drew. So in one experiment, first experiment, we're starting with these 16 guys, right? As a function of time. Do you think they will remain 16? What will happen? First experiment, second experiment, 212 experiments, and it's collecting all that data and making this histogram. Everybody understand what a histogram is, right? Right? How is this being done? So you do experiment number 1, okay? Experiment number 2, and you collect how many are 16, 16, 16, 16, an average, and how many are there? 212. So that's what I plotted here, right? Everybody understands, right? Okay. So what do you think in second generation, third generation? This remains the same. This object does not change, this histogram. What will happen in very long times? That much we know, right? Experts are not allowed to answer. We are all learning, so yeah. By now we're experts. We have seen, yeah. No, there's no fitness. We are in the neutral world. All are equally good. Only stochasticity. That's you have to, what you're doing. Fluctuations. Yeah, that's what we expect, right? What will happen? In the first experiment, eventually, some experiments, in another experiment, in experiment number, I don't know, 204, maybe you got degraded, right? Eventually, that's what is going to happen. That's what we just learned in 2 contexts, right? The coin and this Fisher model, right? So I expect that at long times, I expect that this will become here and here, right? Can you also give me numbers? The height of these peaks. I'm drawing equal, but yeah. Okay, yeah. So that's it eventually, right? But you don't do experiments eventually, right? Of course, you don't wait. Okay, I'm going to take data only at 0 and at 19 generation. You are doing experiments, so much work. So we will, do it at first generation, second generation. All the data we will collect. So let's try to understand not just the eventual, but the dynamics of this. So, okay, let's see if you can guess. So we have to go from there to there. Any guesses? Actually, it's hard, so I will draw it for you. Okay, so this is what he found at t equal to 1. This is what he saw. So 16, then it became something, it reduced in some asymmetric fashion. And here he had just this number is about 40. So this is after this 1 generation. Okay, so that peak of where 212, only about 40 experiments he saw 16 of those mutants of B7 allele. Okay, and then so then he kept on doing it, and then at t equal to 10, t means generation, and t equal to 10, he saw something like this is about maybe 10 here at 0, and again I think it's similar. Maybe it's a little bit higher here. Okay, I'll just draw approximately here. Something like this. Sort of flattish thing. So that peak in between became very flattish. And then at t equal to 19, so this is by the way infinity. So at t equal to 19, we saw this. $40 here and 60 and some stuff in between. Something like that. Okay? Does this make sense? Now that you got used to it a bit. And these are the answers. Do they make sense? 0 we understand, infinity we understand, but this is the dynamics in between what's going on, and that's very informative. Does it make sense? Anybody wants? Yeah. Okay. Want to offer me an explanation? Do you think you would have? Let's talk about this guy. Does this make sense? Right? It's like this. You're not going to stick around at 16, no? right? I mean, this coins flipping, you'll get 52 rupees, 48 rupees, and so on, right? So it'll spread a bit. And it spreads more and more. And then eventually, it becomes this sort of a U-shaped thing. Okay, which is what you guys said, right? Except the numbers are not quite, not 106. It's around 40, 60, and there's some stuff in between. Here, our prediction is that there should not be anything. There should not be any heterozygotes left. We should have only homozygotes, right? That's a prediction. Okay, but that's what he saw because he stopped the experiment at 19 generations. Okay, so it's very interesting, at least for me as a theoretician, that this full answers that what's happening in between, you can actually understand, you can write formulas for it. We will not write it, but I just want to tell you, because I think some of you are theoreticians. So there is a theory called diffusion theory. So for those who work with these things, which is a complicated differential equation, which says the probability that there are X fraction frequency of those heterozygotes of that mutant frequency is X at time t. For this, you can write a complicated equation, which looks like this. I 2 and 4 and I'm not very sure. Okay, whatever these symbols mean, doesn't matter if you don't know this. Okay, but there's some complicated partial differential equation one could write it down. Those who are physicists and work with these things, let me just point out to you. If I did not have X in between, inside the second derivative, then we will get the coin problem, the random walker problem. In this evolution thing, it's very interesting that you get this stuff inside. And that's because of heredity, or you can argue about this. Okay, this is solvable, and answer is pretty complicated, but you can write down an expression for it. And then from this answer, if you plot, if you at least qualitatively, they look very similar to what he saw in the experiment. So that's very nice. Right? Okay, so we can understand this theory using a theory as well. Okay, so but that's a very involved object. One can also look at, because I'm talking about dynamics here, one can also look at heterozygosity. How do you think it's changing? Let me just ask like this. What's the fraction of heterozygotes in the population as a function of time? Initially, everybody's a heterozygote, right? So I'm plotting this H as a function of time and time. Fraction of the frequency of heterozygotes. Again, we know this, and it's very useful to think of limits, extreme things. So we know at t equal to 0, all are heterozygotes, right? So frequency is 1. At very late times, we know there's no heterozygote, right? Because all of them became homozygotes, right? So therefore, this has to become 0 somewhere, right? And the question is, how does it go from 1 to 0? Can you guess, looking at those plots? Huh? Loud? Huh? Yeah, so how does it look? H of t as a function of t. How does it go? Does it increase first, maybe, and then decreases because it has to become 0? Maybe it oscillates. First increases and decreases, okay. Like this. Yeah? Okay. Heterozygosity, it cannot increase beyond 1. So it cannot be increasing. That's the max, right? It's a fraction. Yeah, yeah. Okay. So yeah, so it decreases, and I'll write a small function for you. It decreases what is called exponential. Okay. So it e to the minus t by 2n or 4n. I always confuse this, but something like that. It decays very fast. Exponential functions are the functions which decay very fast, very rapidly. Okay, it's like 1 by 2, 1 by 4, 1 by 8. So that's a decrease, rapid decrease. That's precisely that is. Okay, so they plotted. Question? That's an average, mean heterozygosity. Thank you. So average over all these. Yeah, right. Thanks. Yeah. So then these guys, they plotted, he plotted this, and he wanted to see along his formula. And then he found that he could fit the data pretty well if instead of n equal to 16, his population size 16, right? Instead of 16, he used a number like 10. Okay, so he got a good fit with that. So that n now is called effective population size, and you will hear much more about this from Mike. So get good agreement if between theory and experiment. If you use NE to be about 10. Okay, and the reason being maybe there's something called random mating, which we haven't spoken about, but any of them can mate with another one. Maybe that's not valid, or maybe they're just looking at the neighbors to mate and so on. So there could be various factors, and people try to characterize that. Okay, so but this is some effective population size. If you plug it in that formula, then he got a reasonable match. Okay, so now, okay, so that's one thing. Okay, now let's just come back to this question, that when he stopped the experiment, he stopped at 19, and you got 40 here and 60 instead of 106 each. But of course, you expect that if we had waited long enough, if we had done more experiments, you would wind up that, right? Okay, so we would like to know another question. So we spoke about fixation probability. Another very important question is, what's the time of fixation? So if I know what's the average time would it take for me to reach these states, or here in this, then I would have known how long to carry out this experiment. So I'll write that here. So from this kind of theory, mean time to absorption. Absorption means either all A2s or all A1s. Fixation means only all A2s, or whichever state you choose. So one can write an expression for that. And it turns out, if I plug in everything, turns out to be 2.8, I think. 2.8 NE. And NE, as I said, is 10. So about 28 generations on an average. If we had done experiments from the 10 generations, we would have winded up with something like that. Okay, so that's again comes from a sophisticated theory. I don't know a simple argument to predict that 2.8. Okay, so we have about 20 minutes. So instead of starting very, very new things, let's do something simple. Okay, is it okay with everyone? Yeah. No, there's a general formula. There's a general formula which I didn't know. I'll give it to you since you asked. So I don't know if you can see it or not, but I'll write it for you. Thank you for asking. Log. So it's saying that if you started with a initial frequency and not by n, then the time to reach this kind of state on an average, there's a fluctuation in that, of course, that is given by this formula. Okay? So it's not. By the way, the first thing that we said about coins, do you think, can you guess how many tosses would you need to leave this room? I know the answer. So, okay, let's not go in that direction. Let's go ahead with a little bit more we will do, and then we will stop. Sorry? There's a minus 4 NE because, yeah, minus is there. Yeah. Okay. Okay, so let's do something small, and then I promise to stop. Okay, so this is all the way in which everybody is good and no changes are happening, right? But now we need mutations. Mutations happen all the time, right? Okay, so let's now try to figure out genetic drift. Less mutations. Same kind of model, but now I'll just add a little something in it. So same Wright-Fisher model. Now the only new thing is that A1 can become A2 with some probability mu, And A2 can become, so A1 can become A2 with probability mu and remains A1 with probability 1 minus mu. Similarly, A2 can become A1 with some probability and otherwise not. Okay. So I've introduced these to you, mutation probabilities, and they can be related to mutation rates. Okay. So we just run the same kind of model, and I'll just draw the same way as in my notes. Okay, whatever. Does this look okay to you? This one? Yeah. So I'm saying now A1 can become A2 with some probability. So that's probability is mu. And it does not mutate with probability 1 minus mu because either you mutate or you don't mutate. And A2 can become A1 or not, but maybe in general different probability. That is all I'm saying. So if I now write down Wright-Fisher model, I can get a generation like this. Is this okay with everyone? Everybody understand what I've written here? Make sense? Yeah? Yeah. So this is first generation, zero generation. Yeah, first generation, yeah. Yeah, so parent is A1, and I'm writing A2. Why? Because mutation has happened. So this is the parent, but then it made a mistake, okay? And therefore, the A1 became A2 with a probability mu. Okay? Earlier it was A1, no? Because you just copied. But now there's an error, and therefore it became A2, and similarly here with some probability. These are A2s, but this became A1 because of the mutation. Okay, same question. What's the probability of all becoming A2? Okay, let me ask this question again. Does this question make sense? So suppose we run this program in our heads. Suppose all became A2. Is that the end of the story here? No, because A1 can be generated back. That's what mutation is going to do for you. So everything became monomorphic or same type, but now you're generating the new ones, right? So therefore, now mutations going to create diversity. So drift has this characteristic. It tends to remove drift, deplete drift, diversity, whereas mutation is going to generate it, right? Okay. So therefore, if I now look this program, if you run for a long time, I'm not going to get these two peaks that we saw in Buri's experiment, right? Okay. What do you think we are going to get? What's the long time state of this model? So there are these two competing forces. One is trying to remove, making them A2 because of stochasticity, but then mutation comes back in and generates it back. So some kind of competition, tug of war, some balance may be reached. It's called mutation drift balance. So at long times, and I would like you to guess. So now, without mutations, we had this as a distribution. We had no A2s or all A2s, right? Now with mutation, how do you think the distribution of A2s look like? So let me repeat this and write it better. Let's say there's all this. So run this in your head, please, and see what kind of state we have. Okay, so the question I'm asking you is, at long times, on a plot, the frequency, if you like number, let's do number. number of A2s and the probability of. So when there was no mutation, we saw it was all half, right? Right? That's what we had earlier. Now what happens? How does it change? Okay, so that's something you have to think about. So maybe I think I should just stop here and I would ask you to start thinking. Yeah, I think I'll stop talking. We have 15 minutes. So I will stop talking, take this out, and I can come to you and you can feel free to ask doubts, which you were hesitant to ask. Okay, fine. We'll stop here. And please try to think of this question. And if you don't like this question, think of the first question, which was the fixation probability in expanding population. Size, initial size, not with okay. So let me stop here.